Greetings, fellow researcher. So, my name is Jonathan, and I'm from the University of Zurich. My project is about a molecular perspective on seed development and germination in Tylosema fassoglense, which is commonly known as marama bean. And I'm gonna take around 5-6 minutes to finish to go through everything. So, I want to start giving a brief context, which is about the increase of global population. So the world population growth, it poses a challenge to the agriculture system that needs to ensure high yields every year in order to feed everyone, to meet the demands of um, food production. And And on top of that, climate change also inflicts more pressure into the agriculture with higher temperatures, water scarcity. Uh, all of this leads to crop failures, so we suffer with yield losses, especially because our agriculture relies on just a small number of crops that do not perform well on these conditions. So an alternative for this is the use of underutilized crops or wild plants whose fruits are gathered, uh, offering new avenues for agriculture. And what is it? So underutilized crops are these plants that are not widely grown or commercialized. It doesn't have, don't receive much attention from research or from the economy. Uh, Despite their potential for nutritional, environmental, and/or economic benefits. An example of these underutilized crops is Tylosema fassoglense, as I mentioned in the beginning, marama bean. So, although the seeds of the Tylosema species are of high nutritional value, for example, the protein content of marama bean is similar to soybean, and the seeds they have been it has been gathered for millennia from the population in Africa. So here is the distribution of the whole genus, all species in the continent. None of these species has been domesticated so far. And another great advantage that turns marama into a good option for agriculture is having a high tolerance to drought, heat stress, and poor soils. So their potential cultivation could contribute to a more sustainable agriculture. So my research focus is to better understand the molecular mechanisms underlying seed germination in Tylosema fassoglense, especially because these seeds they require scarification in order to germinate. So these beans they have a hard and thick seed coat, which hinders the process of germination. That's why we are interested not only in the molecular mechanism of the germination, but also in the seed development, so the seed coat formation. And some of the questions that I want to answer is what genes are involved in formation of the seed coat, or what is the effect of scarification on the transcriptome during seed germination. But in order to answer these questions, first I observed the water uptake kinetics during germination to have a better idea on how this process occurs in these seeds. And from this particular experiment, I could I could establish time points for the further experiments. So here you can see in the graph that in the y-axis I have the weight, the seed weight, and in the x-axis the different time measurements. So each point also is an average of a group of seeds, and this was a nice way to have an idea on how these seeds would behave during germination. So, in order to survey the transcriptomic profile during germination, and identify differentially expressed genes associated with the process. Um, the approach I used to investigate it was with three pre-germination treatments, which consisted of gibberelline treatment, escarification, and the control, which were seeds untreated intact in this group. And the escarification was done by wood file and sanding the seed coat of these beans. I collected the 4 time points. These samples are going to be sent for sequencing in the near future, and then we are going to start with the analysis. Beyond that, we are also interested in the seed coat structure of Marella. So here you can see a picture of a split open seed with the zoom for better visualization of the seed coat. So here we have two transversal sections, one normal and the other one is stained with toluidine blue. And with this we hope to have a better idea on how this structure is organized and to understand how it works during the process of germination. So as take-home message for my project, I would like to mention that our work wants to provide molecular insights into the poorly understood germination mechanism of marama bean, and not only that, but also gain genetic knowledge to facilitate future domestication efforts. So identifying candidate genes that could facilitate the process of domestication and breeding in the future, and ultimately increasing food security with marama. And with this I conclude my talk. If you have any question, suggestion or comment, I will be more than happy to discuss it in person. Otherwise, just send me an email and I will be happy to reply it. So thank you, enjoy the conference, ciao!